The learning outcome for this lesson is to simply recall what an electron microscope is. So I'm going to talk a little bit about the electron microscope. So obviously you can see this is one here. It's much bigger than most light microscopes. It's incredibly expensive, so you tend to find it mainly in universities and research institutions. It requires a technician to operate this, so no one can just basically rock up to this machine and expect to use it. It requires a trained technician. Quite costly to use, and there's usually a bit of a queue for you to get on to use this electron microscope. Another difference is you've got your image on an LCD screen, although some of the more powerful light microscopes have exactly the same now. You only see in black and white, and the big difference between a electron microscope, correctly called a transmission electron microscope, is that there's an electron gun here. So rather than using a beam of light, a beam of photons, the electron microscope uses a beam of electrons. So that's the key difference they're expecting you to recall in the exam, that a beam of electrons is used. We've got the specimen port here. So basically you will have what's called a little stub, and on that little stub you'll put your specimen. So your specimen goes on here. It's a really thin specimen because the electrons have got to pass through it, so it's ultra thin specimen. Goes through the specimen port and your specimen will sit here, and the beam of electrons are basically fired through your specimen, and then the electrons hit this fluorescent screen, and that causes an image which can be seen through these binoculars here. So you'll see your image. So the two key differences that you need to recall in the exam about electron microscopes, and the first one is with regards to magnification. Now, depending on what source of information you read, you'll probably get different answers, but by and large the magnification can be 10 million times. So that's an extraordinary magnification. But what's more important than that actually is the resolution that you have with an electron microscope. So the resolution here is 50 picometers. In other words, this allows you to see in much greater detail. For us, thinking about what's inside a cell, so we can see the structures, the organelles or substructures inside a cell, the ones to recall would be the ribosomes. So the ribosomes are tiny little dots, even these are too big. So ribosomes are really small. There's no chance of you seeing a ribosome with a light microscope, but you can see them with the electron microscope. Another structure that you can see with the electron microscope would be the mitochondria. So you can see the mitochondria in much more detail. Here are a couple of photographs taken with the electron microscope. These are called micrographs, of course. Perhaps it might be called an electromicrograph. And this is a mitochondria, and you can see these tiny little structures in here known as cristae. You'll get to that when you encounter A-level biology, should you head that way. And these are the chloroplasts with the stroma and the grana inside. So it's quite possible with an electron microscope to actually look at these in even more detail again.